Alright, this is Macromolecules Part 2, just proteins and DNA, or nucleic acids. Proteins are molecules that consist of one or more polypeptide chains that are folded and coiled into different conformations. Polypeptide chains are just a polymer of amino acids, and I'm going to always abbreviate that AA, that are arranged into a specific linear sequence and are linked by peptide bonds. Here you can see a polypeptide chain, right? We're going to talk about that again in a minute. Sorry, hang on. All right. Proteins are very abundant and make up to 50% of cellular dry weight. They're important as structure, storage, transport, signaling, response to stimuli, movement, defense against foreign and disease-carrying organisms, and as catalysts of biochemical reactions. They vary in structure. Each has a unique 3D shape called the conformation. Right. Conformation. And each is made up of only 20 out. Let's trip over my words today. You only have 20 amino acids that make up all of our proteins. It's just a matter of them being in a different order, mainly, just like letters of the alphabet, different lengths, different orders. Most amino acids consist of a asymmetric carbon covalently bonded to a hydrogen atom. Oh man, hang on. Hydrogen atom, a carboxyl group. Let me just draw the whole thing. Carboxyl group, amino group, and then a variable group. And that's specific to each amino acid and gives the amino acid the physical and chemical properties. Each side chain determines the unique properties of each amino acid. Let's see. Some amino acids are polar, some are non-polar. Thank you. If they're polar, they can be charged or uncharged. So polar is just going to mean hydrophilic, and then acidic or basic. They are linked by peptide bonds, which are covalent bonds formed by condensation reactions, and they link the carboxyl group of one amino acid to the amino group of another amino acid. The protein function depends on shape. Protein conformation is the 3D shape of a protein. And then the native... Not right now. Thank you, though. Native conformation is the functional conformation of a protein found under normal biological conditions. You have 4 levels of protein structure. Primary structure, I'm going to abbreviate that as primary, is the unique sequence of amino acids in a protein. It's determined by genes. Any slight change can change the conformation, and that happens with sickle cell disease. It can be sequenced in a laboratory and now by computer. Frederick Sanger sequenced insulin in the 40s and 50s. So that's primary structure. Next up you have secondary structure. And you have two possibilities here. You can have the alpha helix or beta pleated sheet. This is still going to contribute to protein's overall conformation. It's stabilized by hydrogen bonds between the polypeptide and protein backbones, and between the carbonyl and amino groups. Tertiary structure, let's go green. Tertiary, third order, is irregular contortions of a protein due to bonding between the side chains. This is side chains. You can have weak interactions that help to stabilize hydrogen bonds, ionic bonds, and hydrophobic interactions. Or you can have covalent linkages like disulfide bridges. These are a big deal. Disulfide bridges. And they occur between two cysteines. Monomers that are brought together in the folding of the protein, and it reinforces the conformation. And then quaternary structure is here. And it results from interactions between several polypeptide chains. Let's put, these are between the actual polypeptide chains. So like hemoglobin, there's 4 different chains that have to fold together to make the protein. Denaturation is the process that changes, and I have a picture up here. I know this is a mess. I promise all videos won't be this way. You have your lovely protein, what it's supposed to look like, and then either by heat or pH or something, the protein becomes denatured and inactive. Sometimes that is reversible. Enzymes, enzymes are an important protein group. They help to change the rate of chemical reactions, and we'll talk about them more in a couple of weeks. With nucleic acids, there's 2 types, DNA and RNA. DNA carries all your genetic information. It's copied and passed from one generation of cells to the next. It's primarily found inside the nucleus, makes up genes, and it has instructions for mRNA and protein synthesis. RNA is responsible for the synthesis of a protein. The site of protein synthesis is the ribosome in the cytoplasm. And there's something, the flow, that is not going to work here. The flow of genetic information is going to go from DNA to RNA, or mRNA to be more specific, to make a protein. When we talk about DNA as a polymer, it is made up of nucleotides, which a nucleotide is a sugar, a phosphate, and a base. In DNA, the sugar is deoxyribose. In RNA, it is ribose. The sugars and phosphates make up the backbone of DNA, and the nitrogenous bases form the little rungs. When you talk about the bases, there are 4 types. If we talk about DNA, we have adenine, guanine, cytosine, and thymine. RNA, we substitute uracil for thymine. You can see pyrimidines are 1 ring and purines are 2, and purines and pyrimidines are the ones that bond. So G to C, A to T. Nucleotide functions, monomers. We can talk about ATP here real quick. ATP is cellular energy, and it will change the nucleotide functions from just being, or change our function here from just being pure genetic information to be the exchange of energy as well. Nucleotides are joined by phosphodiester bonds. And then with DNA structure and shape, it is a double helix. And the first guys to figure that out and build it are, we're just going to go last names, Watson and Crick. The 2 strands are complementary and serve as templates for making new strands. Here the blue strands are old, the orange strands are new. We're going to talk about this more later. And that is finally the end.